is cell counting. Cell counting is very, very useful because sometimes you like over here on this plate, you'll get a bunch of colonies, or sometimes you'll get, actually most of the times you'll get more than that. And counting them all by hand can be tedious, especially when it gets like 500. So ImageJ does a really nice job of it. So open up your plate image. Open with. That pop up. Okay, so kind of how Anthony showed in the beginning, we're gonna end up cropping this because there's a lot of background that we just don't want, and we want to make sure we're kind of going not to the edge of the plate, but like to the edge of, I guess, the agar, because we don't want, what ImageJ will do is sometimes it'll pick up like those lines, and that's just not what we want. So yeah, just like kind of play around with it depending on your plate, depending on how your picture is. Like I have a little glare here, so that's not always great, but then you're gonna go to edit, clear outside. So now it's like, okay, this is just my agar, this is just where the colonies are. Sorry, show the directions as well. Okay, then we're gonna go to image and we're gonna change back to 8-bit. You're always gonna wanna work in 8-bit just because having RBG is just gonna get a little harder to analyze. As everything should be binary, it's much better. And then also, like, again, adjust your brightness and contrast. The more you have contrast of, the better it is. It's easier to see and create distinctions between what's like important and what's not. Okay, so now we're going to threshold these, and just to help again to like make sure everything's binary. So like our cells are going to be white and the background is just going to be black. So we're going to threshold. And so I like using red just because it's a little easier to see. So right now you can see like my background. So whatever is red is kind of like what you're going to focus on. So like right now my background's red, and sometimes this will happen. So what you want to do is when you go back, you can like say edit, invert. So now it's back to being red, and then you can adjust your threshold again. So then you kind of just want to play around with it. So like this is not great because now I'm like, my colonies aren't segmented. I have a big red, like that's just not what we want. So you have to find like a happy medium to minimize all these red spots, but while you're still trying to get those small little colonies right here. So obviously it's not perfect because like you can see there's some on the left-hand side that are just, that's gonna mess it up. So if you want, you can now like go back again, and because it's a little bit easier to see what you really want to focus on, and you can like recrop if you'd like to do that. So I'll just recrop just a tiny bit. Just until you're like satisfied or you know you're getting enough of what you want from the image. So yeah, so like I'm trying to just exclude that. Clear outside. Okay. So that seems pretty okay to me. So I'll apply it. And now as we can see, okay, can we move this? Okay, as we can see over here, we have our cells are white and our background is kind of black. So so ImageJ is gonna just count all the white spots in it. So now there's something that you could do if your plate kind of looks like, if you have, let's say like a clump of colonies or something, like I guess right here, if these two colonies were together. You can go to process, binary, and then watershed. And what this will do is that it'll kind of separate those colonies and make small little segments. But the problem is I kind of have a band here, and my colonies are more or less kind of separate. So I'm not really going to do that with this image. So now we're going to go to analyze, and we're going to say analyze particles. So you're going to want to make sure you have display results, clear results, and add to manager. Those are like just the main important things. And then this is the thing that you're going to want to play around with is your size. So what I like to do is how Anthony kind of showed in the beginning is like measure the area of like your smallest colony. So I did that before, and that's like 175. And then sometimes if you have like a big colony that you know is, okay, that's a separate bacteria, I don't want it, you can kind of exclude it that way. But I do have some big colonies and I really don't care that much, so I'll just go to infinity, it's fine. And you say okay. So now you're getting a cell count right here. It says a cell count of 90. That's like your summary page, so if you don't tick that, you won't get that back. And then you have another results page, which is like the area of all the sections that it started to find and all like the colonies, I guess it counted. And so this is like a really good estimate because I believe the colonies were like 91 or 92 when I individually hand counted it from my lab. So it's not perfect, but it's still like much better than me counting 90 colonies. So, or if it gets to like 500, it doesn't take that long to do this. So that's fun. And then if you want to put it in a CSV file, all your data will get exported into a CSV file if you if you want to save it. Okay. Oh, okay. So what the ROI manager does is that it overlays it so you can actually see like what it's counting as a colony. You can zoom in if you want. Okay. Well, okay, so you can like zoom in, and so let's say 175 wasn't like a great number. Maybe I needed to go a little bigger, a little smaller, then I can see exactly what colonies it's trying to count and then adjust it that way as well. So yeah.